Hello, my name is Amilla.
And my name is Anzhelika.
We are students from Almaty, We are currently studying in Thomas Education School, and today we would like to share our research on the isolation and characterization of the Demidova fundus, Archaeopteryx oligospora, for biocontrol of nematodes. And for the introduction part of our presentation, I would like to pass it on to my research friend, Anzhelika.
So, phytoparasitic nematodes are one of the most serious threats to agriculture, causing significant crop losses and lowering quality of agricultural products by damaging the roots of the plants. With the growing global demand for food and increasing pressure on the agricultural sector, finding effective and environmentally safe methods to combat this pest is becoming increasingly urgent. Archaeopteryx oligospora, known for its ability to capture and destroy nematodes using specialized trapping structures, as you can see here. On this graph is illustrated interaction among oligospora, nematodes, and plants. As you can see, nematodes are basically parasites that infect plants and harm their health, and plants are one of the hosts for nematodes. Oligospora attract, recognize, trap, and destroy nematodes, thus promote plant growth. Demidova fungi offer a promising solution for biological pest control, minimizing the need for chemical pesticides that can negatively affect the environment and human health. This scientific novelty on this research lies in the comprehensive approach to studying the Demidova fungus Archaeopteryx oligospora under the specific conditions of Kazakhstan. Despite the global interest in this organism, its ecological and biological characteristics are under-examined in our country. The practical significance of this research is in developing effective and environmentally safe methods of controlling phytoparasitic nematodes in Kazakhstan. Now, my research partner will explain you about our methods and results.
Yes, thank you, Anzhelika. Let's move on. So the next step of our presentation is the methodology. First step is soil sample collection. We collected soil samples in our local area by using the envelope method. And then the next step was the investigation and isolation of soil predator fungi by surface cultivation. That cultivation was done on 4 different types of agar layers. So the third step is the identification of isolated predator fungi. It was made with the help of the Dodgenton key, and we also sent the materials to the biotechnology laboratory to get the results. The next step is the isolation and counting of nematodes. We were able to conclude results with the help of Bertrand's final method. So in the methodology, the last 2 steps are the most crucial because they are the experiments. This experiment, determination of nematophagous activity of predatory fungi in vitro, is the experiment in artificial environment, so in man-made environment. And the last experiment is the evaluation of the impact of predatory fungi against nematodes in the tomato rhizosphere. It is experiment in vivo. In natural environment with plants. Let's move on. These are the cultivation services that I talked about in the methodology. You can see here that the cultivation results differ vastly. We can clearly see that the wort agar had the most growth, and you would think that the most growth is the best option for our research, but that's not the truth. Because apart from the predatory fungi that we need, other fungi also grew, and it was harder for us to extract PF. So instead, we concluded that the water agar that has little to no nutrients was the best option for us. Here's the table of the vivo experiment that I mentioned earlier. Let's focus on the length of the stem. Let's see the results of the control group with the normal healthy soil. You can see that we can group approximately 21 centimeters, and the main group with predatory fungi grew roughly the same. However, we can see a big negative difference when nematodes are added to the soil. It is almost the half of what is supposed to grow. And then we can see that in the soil where nematodes and predatory fungi are added, the fungi is able to get nearly back to normal. And so the same trend is observable among all the other statistics of the plant. Now, that was it for the methodology and results. It's time for the conclusion. Anzhelika, please.
Thank you. To conclude, during the investigation and isolation of soil predatory fungi through surface cultivation, pure strains of Astromotris allegospora were obtained. It was determined that the optimal growth conditions for the strain are from 20 to 25 Celsius degrees and pH level from 6 to 7. The nematofungus activity of the isolated Astromotris allegospora strain was tested. In vitro experiment demonstrated the elimination of up to 80% of nematodes. And in vivo experiment in the tomato rhizosphere showed positive results of the methoplants, demonstrating higher stem length, leaf number, and higher average root and stem mass. That was the end of our presentation. Thank you for your attention.
Obrigado. Hope we will meet you in person.